Moving forward with the study of the secretory pathway, we go on now to study the Golgi apparatus. And in relation to the Golgi, we will first talk about the organization of the Golgi in the cell. Then we will be talking about the way proteins traffic between the endoplasmic reticulum and the Golgi, and what happens at the level of end-link glycosylation in the Golgi. Then we will be referring to lipid synthesis, because an important part of lipid synthesis actually takes place in the Golgi apparatus. And finally, we will be talking about the export of proteins from the Golgi to other locations within the cell. And at this level, we will be referring very briefly to vesicular traffic in the cell. Vesicular traffic, however, will be dealt with in another video. Let's start with the organization of the Golgi apparatus. The Golgi is composed of flattened, membrane-enclosed sacs, also known as cisternae, and their associated vesicles. One very interesting feature of the Golgi is its polarity. The Golgi has a cis phase, which is the region of the Golgi that is in closer proximity to the nucleus, as well as a trans phase, which is the region of the Golgi that is closer to the plasma membrane. The cis phase constitutes the point of entry for proteins into the Golgi. That's the place where the proteins that are coming from the endoplasmic reticulum will be entering the Golgi apparatus. On the other hand, the trans phase is the exit point for the Golgi apparatus, and from there, proteins will be targeted to different compartments within the cell. Between the endoplasmic reticulum and the Golgi apparatus, you find an intermediate compartment known as the Golgi intermediate compartment, also known as the ERGIC for the initials of this compartment. The Golgi intermediate compartment is formed by the fusion of the vesicles that are produced from the endoplasmic reticulum, as shown in this figure. The flattened sac that is produced by the fusion of these vesicles is believed to then move forward in the secretory pathway, therefore producing the cisternae of the cis Golgi network. As this cisternae moves forward in the secretory pathway, it generates in turn the medial and the trans Golgi network. The vesicles that are then generated from the trans Golgi network take the proteins that have reached this location in the cell to different locations within the cell, including the plasma membrane, as well as endosomes and lysosomes. Let's now address the question of how is protein traffic between the and the Golgi apparatus taking place. As previously indicated, protein traffic from the endoplasmic reticulum to the Golgi takes place in vesicles that bud off the membrane of the endoplasmic reticulum and that are destined to go to the ERGIC, the Golgi intermediate compartment. The obvious question on this process is whether there are specific sequences that will target proteins to go from the to the Golgi. And the answer to this question is very simple. For membrane proteins, there are specific sequences that serve as targeting sequences to direct proteins from the endoplasmic reticulum to the Golgi. Those sequences are the so-called dihydrophobic sequences and diacidic sequences. And those sequences are always located in the cytosolic domain of integral membrane proteins. So these are proteins that have a transmembrane domain, as shown here. All of these have transmembrane domains. They also have an luminal domain, and then they also have that cytosolic domain, which contains the targeting signal. While integral membrane proteins of the contain specific sequences that target them to the Golgi apparatus, soluble proteins, that is, the ones that are not anchored to the membrane, soluble proteins seem to not have any specific targeting signal that will direct them toward the Golgi apparatus. Therefore, the traffic of soluble proteins from the ER to the Golgi seems to follow a default pathway. That is, all soluble proteins that are present in the ER will by default go into the vesicles that target proteins to the Golgi apparatus. The existence of this default pathway implies there will be some proteins that will end up going to the Golgi, but that should in fact remain in the endoplasmic reticulum. These proteins, therefore, must contain some sort of sequence that will allow them to be retrieved from the Golgi back to the endoplasmic reticulum. Currently, there are two different types of retrieval sequences that have been very well characterized. The first one is the KDEL targeting signal, and this signal, the KDEL signal, is located at the C-terminus of soluble proteins that are destined to remain as soluble proteins in the endoplasmic reticulum. The second signal is the KKXX signal, which is located at the C-terminus of integral membrane proteins that are meant to remain in the endoplasmic reticulum. Now that we have already talked about the signals that target proteins to remain in the ER or to go to the Golgi, now we can talk about the processing of N-link glycosylation in the Golgi apparatus. We had already mentioned how N-glycosylation takes place during the co-translational translocation of proteins into the endoplasmic reticulum. When proteins are let go out of the endoplasmic reticulum, the initial 14 sugar residues that were simultaneously transferred during the initial N-glycosylation has been trimmed down to only 11 sugar residues. And remember, the trimming and addition of one extra glucose to this N-link glycosylation site in the protein is important for the proper folding of the protein, acts as a mechanism of quality control to ensure that the protein is properly folded. So by the time that the proteins are let go from the ER, they have 11 sugars attached to them. When the proteins arrive to the Golgi with those 11 sugars attached to them, the sugars now are going to be undergoing extra steps of processing. First, a few mannose residues are taken out, and then other sugars are added in sequential steps to the mannose residues, therefore providing for further complexity to the sugars that are added to the protein. The process ends with the addition of sialic acid to the outermost sugars in the glycosylation site. Sialic acid is a critical element then that will provide a different chemical nature to the sugars that have been added to the protein. Sugars lack charge, that is, they are not normally charged under the natural pH that prevails in the cell. However, sialic acid is normally under normal pH conditions negatively charged, and coating the sugars with sialic acid molecules will therefore provide a negative charge to the sugars that are coating the protein. And overall, the addition of sugars that are then covered with sialic acid are then going to provide a net negative charge to the proteins that have been N-glycosylated. The addition of sugars to some proteins during their traffic in the Golgi apparatus has the additional function of allowing some specific proteins to be targeted to the lysosome. And the signal that allows proteins to be targeted to the lysosome is the addition of a phosphate group to a mannose residue. Obviously, not all proteins that have been N-glycosylated will receive that particular phosphate group in that mannose residue. So then the question becomes, so how come that some proteins will receive that phosphate group whereas other proteins will not? The hypothesis that has been postulated to explain why some proteins receive that extra phosphate group is the existence of what has been called a signal patch in the proteins that need to go to the lysosome. That signal patch allows for some specific enzymes to then add the phosphate residue to the mannose. But then the question is, what is that so-called signal patch? And to this day, nobody really knows, so go figure. Now on to talking about lipid metabolism in the Golgi. We had previously mentioned that all phospholipids that are derived from glycerol are synthesized in the endoplasmic reticulum. There is another important precursor for phospholipids that is also synthesized in the endoplasmic reticulum, that is ceramide. Ceramide arrives in the Golgi, and in the Golgi it is modified and converted into either sphingomyelin or glycolipids. Sphingomyelin is a phospholipid, and in fact it is the only one that is not synthesized in the endoplasmic reticulum. It is also the only phospholipid that is not a glycerol derivative. The synthesis of glycolipids is a little bit more complex simply because it requires the addition of several different sugars to the initial precursor. The initial addition of glucose occurs in the cytosolic side of the Golgi apparatus. After the addition of the initial glucose, this glycolipid is then flipped to the other side of the membrane so that it ends up facing the luminal side of the Golgi, and then other sugar residues are added, therefore producing the mature glycolipids. However, those additional extra sugar residues that are added then prevent further flipping of these glycolipids. And the resulting effect of this is that glycolipids are located only facing the luminal side of the Golgi, and later on, as the circulatory pathway progresses, they end up facing the extracellular side of the plasma membrane. And this is an important characteristic that you should remember from this moment on. Both glycolipids and sphingomyelin are located only on one phase of the lipid bilayer, and that is the extracellular phase, which is the equivalent of the luminal side of the Golgi apparatus. The final stage of the circulatory pathway is the export of proteins from the Golgi apparatus to other destinations within the cell. Proteins leaving the Golgi apparatus can have different destinations within the cell. On one hand, those proteins can go through the constitutive circulatory pathway and be secreted out of the cell. This pathway is considered to be the default pathway for proteins that are freely floating in the Golgi apparatus, that is, soluble proteins, proteins that are not anchored to the membrane. An alternative pathway is the one of regulated secretion, which leads proteins to vesicles that will fuse with the plasma membrane only when very specific signals are received by the cell. There are numerous examples of proteins that follow this regulated secretion pathway. Among others, there are hormones in endocrine cells. There are digestive enzymes that leave pancreatic acinar cells that are also following this regulated secretion pathway. And finally, perhaps the most important example of this type of regulated secretion is the release of neurotransmitters from neurons. One extra level of complexity of this final stage of the circulatory pathway is added by the fact that there are specialized epithelial cells, such as those that are located in our intestines, for which the plasma membrane has different specialized locations. For instance, there is the apical membrane of the plasma membrane, which is the side of the epithelial cells that is facing the lumen of our intestines. That apical side of the membrane is substantially different from the other sides of the plasma membrane. There is, for instance, the basolateral membrane, which is the one that is then facing the connective tissue that surrounds the basal region of the cells. And then there are the tight junctions that are formed between adjacent cells in those epithelial tissues. Those tight junctions have substantially different protein composition from both the apical membrane as well as the basolateral membrane. And then a logical question is, how can the cell then send proteins to different locations in the plasma membrane? That is, what are the kind of signals that are recognized to direct proteins either to the apical membrane or to the tight junctions or to the basolateral membrane? And finally, to complete our study of the circulatory pathway, it is important to keep in mind that besides proteins being secreted out of the cell, there is also another pathway that takes proteins from the outside of the cell into the inside of the cell, which allows us to introduce two new terms. One of them is exocytosis, which is the secretion of proteins out of the cell, and the other term is endocytosis, which involves bringing in new content into the cell. Exocytosis and endocytosis are part of the same process. And in fact, many proteins that are targeted to the plasma membrane by exocytosis are then recycled back into the cell by endocytosis. Numerous proteins that work as receptors in the plasma membrane are in fact continuously recycled through this pathway. And the place where recycling takes place within the cell is the early endosome. Most proteins and other components that enter the cell by endocytosis are then targeted to the late endosome, and from there they are then targeted to the lysosome. The lysosome is a very specialized organelle within the cell that is characterized by having a very low pH. And this pH is important for the function of this organelle because this organelle is the main recycling center within the cell. The enzymes that are contained within this organelle has the ability to degrade basically every type of macromolecule that can be present within the cell. For instance, there are enzymes that can degrade nucleic acids, and those are nucleases. There are enzymes that can degrade any type of protein, proteases. There are enzymes that can degrade any sugar, those are glycosidases. And there are of course also enzymes to degrade lipids, phospholipids, compounds that contain other type of components such as sulfates, as well as phospholipases, which will be degrading phospholipids. So lysosomes constitute the main recycling center within the cell. All of these enzymes are active only under the low pH conditions that prevail within the lysosome. By the way, this low pH that is present within the lysosome is achieved by the presence of a hydrogen pump that pumps in hydrogen ions into the lysosome, therefore decreasing the pH, lowering the pH within this organelle. And with this, we finish our study of the circulatory pathway. Watch the movie as many times as you have to.